In this video, optical scatter microscopy is used to quantify mitochondrial fragmentation during apoptosis. Although optical scatter microscopy does not normally require fluorescence, in this protocol fluorescent images will be acquired for validation purposes. Following setup of the imaging system, cells are labeled with mitotracker green and treated with staurosporin to induce apoptosis. Optical data are acquired to detect changes in the orientation of cellular organelles. Within 2 hours, the onset of mitochondrial fragmentation can be measured using the optical scatter data, which shows a decrease in the degree of orientation of subcellular particles. The data are then validated by correlating the optical scatter data with the fluorescent regions containing mitochondria.
Hi, I'm Nada Boustany from the Biooptics Laboratory in the Department of Biomedical Engineering at Rutgers University.
I'm Pasternack from the Biooptics Lab.
And I'm Jing-Yi, also from Biooptics Lab.
Today we'll show you a procedure for quantifying subcellular morphological dynamics using a method that does not rely on fluorescent dyes.
We use this technique in our laboratory to study dynamic changes in subcellular structure during apoptosis or programmed cell death.
So let's get started.
Begin this procedure by sterilizing gloved hands with 70% ethanol. Then place a prepared 100 nanomolar stock solution of mitotracker green and pre-warmed BAEC cell culture medium in the hood with the light off and sterilize all surfaces. Next, place a 6-well culture dish with cells plated on cover glass under the hood. Using a Pasteur pipette connected to a vacuum line, remove the cell medium from the wells. Then immediately add 2 milliliters of the labeled medium to each.